Hello, my name is Julio Asaf. Welcome to my video on ossification. Ossification is the forming of bones. Before we get started, let's briefly go over the skeletal system and some terminology. The skeletal system has 5 primary functions: support, storage, blood cell production, protection, and movement. Bones are classified according to shape and structure. Bone or osseous tissue is supporting connective tissue that contains specialized cells and matrix consisting of extracellular protein fibers and ground substance. The distinctive texture of bone results from the deposition of calcium salts within the matrix. The remaining third is dominated by collagen fibers. Osteocytes and other cell types make up only 2% of the mass of a bone. The typical human skeleton contains 206 major bones. They have 4 general shapes: long, short, flat, and irregular. Long bones are longer than they are wide, whereas in short bones these dimensions are roughly equal. Examples of long bones are bones of the limbs, such as bones of the arm and thigh. Short bones including the bones of the wrist and ankles. Flat bones are thin and relatively broad, such as the parietal bones of the skull, the ribs, and shoulder blades. Irregular bones have complex shapes that do not fit easily in any other category. Examples include the vertebrae of the spinal column and several bones of the skull. The typical features of a long bone are shown on the humerus. A long bone has a central shaft or diaphysis that surrounds the central marrow cavity containing bone marrow, a soft fatty tissue. Now let's talk about the cells in a bone. Although osteocytes are most abundant cells in bone, other cell types are also present. These cells, called osteoclasts and osteoblasts, are associated with the endosteum that lines the inner cavities of both compact and spongy bone and within the cellular layer of the periosteum. 3 primary cell types occur in bone. Osteocytes are mature bone cells. Osteocytes maintain normal bone structure by recycling the calcium salts in the bony matrix around themselves and by assisting in repairs. Osteoclasts are giant cells with 50 or more nuclei. Acids and enzymes secreted by osteoclasts dissolve by the bony matrix and release the stored minerals through osteolysis. This process helps regulate calcium and phosphate concentrations in body fluid. The last is osteoblasts. Osteoblasts are the cells responsible for the production of new bone, a process called ossification. Osteoblasts produce new bone matrix and promote the deposition of calcium salts in the organic matrix. At any given moment, osteoclasts are removing matrix and osteoblasts are adding to it. When an osteoblast becomes completely surrounded by calcified matrix, it differentiates into an osteocyte. Now let's get to the good stuff. Intramembranous ossification. It begins when osteoblasts differentiate within embryonic or fetal fibrous connective tissue. This type of ossification normally occurs in the deeper layers of the dermis. The osteoblasts differentiate from connective tissue stem cells after the organic components of the matrix secreted by the stem cell becomes calcified. The place where ossification first occurs is called ossification center. As ossification proceeds and new bone branches outward, some osteoblasts become trapped inside bony pocket and change into osteocytes. Bone growth is an active process, and osteoblasts require oxygen and a reliable supply of nutrients. Blood vessels begin to grow into the area to meet these demands, and over time become trapped within the developing bone. At first, the intramembranous bone resembles spongy bone. Further remodeling around the trapped blood vessel can produce osteons typical of compact bone. The flat bones of the skull, the lower jaw, and the collarbones form this way. Now on to endochondral ossification. Most of the bones of the skeleton are formed through endochondral ossification of the existing hyaline cartilage. These cartilages develop first. They are like miniature models of the future bone. By the time an embryo is 6 weeks old, the cartilage models of the future bones begin to be replaced by true bone. Let's go on to step number 1. Endochondral ossification starts when chondrocytes within the cartilage model enlarge and the surrounding matrix begins to calcify. The chondrocytes die because the calcified matrix slows the diffusion of nutrients. Step 2. Bone formation first occurs at the shaft surface. Blood vessels invade the pericardium, and cells of its inner layer differentiate into osteoblasts that begin producing bone matrix. On to step 3. Blood vessels invade the inner region of the cartilage, along with migrating fibroblasts that differentiate into osteoblasts. The new osteoblasts form spongy bone within the center of the shaft at a primary ossification center. Bone development proceeds towards either end, filling the shaft with spongy bone. Step 4. As the bone enlarges, osteoclasts break down some of the spongy bone and create a marrow cavity. The cartilage bone does not completely fill with bone because the epiphyseal cartilages, or epiphyseal plates, on the ends continue to enlarge, increasing the length of the developing bone. Although osteoblasts form the shaft continuously invade the epiphyseal cartilages, the bone grows longer because new cartilage is continuously added in the front of the advancing osteoblasts. Step 5. The center of the epiphysis begins to calcify. As blood vessels and osteoblasts enter these areas, secondary ossification centers form. The epiphyses eventually become filled with spongy bone. A thin cap of original cartilage model remains exposed to the joint cavity as the articular cartilage. At this stage, the bone of the shaft and the bone of each epiphysis are still separated by epiphyseal cartilage. As long as the rate of the cartilage growth keeps pace with the rate of osteoblast invasion, the epiphyseal cartilage persists and the bone continues to grow longer. That's ossification.